Hello everybody, I am Zeineb from Morocco. I obtained my PhD in immunology at the Faculty of Sciences and Techniques of Tangier, the University Abdelmalek Essaâdi in Morocco. First, I would like to thank the Biosciences Initiative to let me this opportunity to present you part of my thesis work about anti-CENP-B auto-antibodies in Moroccan patients with scleroderma. So let's start. What is scleroderma? Scleroderma or systemic sclerosis is a rare autoimmune disease of the connective tissue affecting many organs including skin, arterioles and microvessels and leading to the development of fibrosis and vascular obliteration. It affects preferentially women more often during and after their childbearing age with a female-male ratio 3:9. And it is frequently detected at an advanced stage due to diagnosis difficulties. Its epidemiology is not definitely established due to the relative rarity of the disease, the difficulty in diagnosis, as well as its extreme clinical variability. Several factors like genetic, immunological, and environmental factors seem to contribute to the expression and heterogeneity of the clinical and immunological characteristics of the disease. Scleroderma is associated with dysfunction of the immune cells, fibroblasts, and endothelial cells, and it is characterized by an unknown etiology and the production of antinuclear and anti-centromere autoantibodies. So, during this disease, there is a production of several autoantibodies, including ACA or anti-centromere autoantibodies directed against centromere proteins. This is a molecular organization model of vertebrate kinetochore. So this model emphasizes the protein-protein interactions and does not necessarily represent their actual localization. To date, more than 60 proteins have been identified as putative components of human centromeres, and a few of these were identified as autoantigens in a number of clinical studies. In 1980, Mouna et al. First identified ACA in scleroderma patients by indirect immunofluorescence analysis, and since then, ACA have been shown to be highly specific for scleroderma, particularly for the limited cutaneous form of the disease. So, the major centromere proteins detected by ACA are CENP-B, CEN-A, and CEN-C. So, among these three proteins, CENP-B is considered as the major centromere autoantigen recognized by more than 95% of scleroderma patients with ACA positive, since high levels of autoantibodies directed against CENP-B have been detected in sera of scleroderma patients with positive ACA. As well as CENP-B is strongly associated with pulmonary arterial hypertension, one of the main causes of death due to scleroderma. In Morocco, data about scleroderma remains scarce due to the rarity of the disease, the difficulty in diagnosis, and the fact that this pathology is not widely known by the population. So, epidemiological studies regarding prevalence and incidence does not exist in the national scale, and only one retrospective survey about the prevalence of anti-centromere autoantibodies in Moroccan scleroderma patients has been published in 2007. Thus, this work aims to study for the first time in Morocco the immune response against the CENP-B antigen by analyzing the reactivity of scleroderma sera against the amino terminal and carboxy terminal expressed domains of the CENP-B protein. So, how the study was done? Our study includes 30 patients diagnosed with scleroderma in the internal medicine department at the University Hospital Center Ibn Sina of Rabat during the period from December 2011 to December 2013. The diagnosis was reached based on clinical and serological manifestations or characteristics of the disease according to the classification criteria of the American College of Rheumatology by an internist doctor after elimination of differential diagnosis. So, there were 24 females and 6 males with a female-male ratio 4:1, and the mean age was 44 years old, ranging between 28 and 54 years. So, at the Laboratory of Autoimmunity in the National Institute of Hygiene in Rabat, we have performed an indirect immunofluorescence assay in order to assess the presence of ANA reactivity. So, all sera were tested on commercially available Epto cells, and sera which revealed positive by immunofluorescence and have homogeneous nuclear or speckled pattern were tested by ELISA to look for auto-antibodies to extractable nuclear antigens. The tests of ELISA and immunofluorescence were performed on an automated system, Biorad PhD system. Sera from 11 patients with scleroderma and ACA positive were tested by immunoblotting analysis at the Laboratory of Biomedical Genomics and Oncogenetics research. So, this sera were tested by immunoblotting analysis using as antigens the purified amino terminal and carboxy terminal region of recombinant CENP-B protein. Firstly, the purified recombinant of CENP-B fragments were separated by SDS page in many gels and transferred to a nitrocellulose membrane, which were then incubated in blocking solution for 1 hour, and afterwards each serum was incubated with the membrane in TBS twin for 1 hour, and then the membrane was washed 3 times in TBS, and then incubated for 1 hour with the secondary antibody anti-human. Finally, the membrane was washed again, and the colorimetric reaction was developed with chloronaphthol for 10 minutes. So, among the most among the most frequently observed manifestation in our series, we found Raynaud syndrome in a half of our patients, followed by sclerodactyly and arthritis in 30% of our patients, and skin sclerosis in 23% of patients. So 6 of these patients had Raynaud syndrome associated with sclerodactyly and arthritis, and these clinical manifestations were more frequent in women than men. The test of immunofluorescence revealed four fluorescence patterns: the homogeneous, speckled, nucleolar, and the centromere protein, the centromere pattern. This latter is the most found in our series, and it has been described in 11 patients. As you can see here, we have a picture of indirect immunofluorescence on EP2 cells performed with scleroderma serum and demonstrating a centromere positive pattern. So, in order to study the anti-CENP-B autoimmune response, these 11 ACA-positive sera were tested by Western blot analysis against the expressed amino-terminal and carboxy-terminal CENP-B protein. The centromere antigens both migrated at the predicted molecular weight for amino-terminal and carboxy-terminal CENP-B, as shown in this image of comacysteinine gel of expressing amino-terminal and carboxy-terminal region of CENP-B. So, as shown in each panel of from left to right, we have molecular weight markers, carboxy-terminal CENP-B and amino-terminal CENP-B respectively. In this image, we showed the Western blot results of scleroderma sera against the two domains of CENP-B protein, and the reactivity of scleroderma sera against the amino-terminal domain of CENP-B are shown in this line, in line 1. So, among the 11 patients with ACA-positive, reactivity against the amino-terminal CENP-B protein was observed in 6 patients. Our results are in accordance with the Spanish study showed that the amino-terminal region of CENP-B harbors more reactivity as an autoimmune epitope than the carboxyl-terminal domain. However, none of all sera tested in our work reacted against the carboxy-terminal domain of the CENP-B protein. In conclusion, we can say that our finding showed for the first time in Morocco that the amino-terminal CENP-B autoantigens was the dominant autoepitope in our series. These results may have a great value in the identification and selection of patients requiring a follow-up and an appropriate treatment. The identification of the centromere antigenic targets may contribute to better understand the ANA response in scleroderma patients and could be beneficial for the diagnosis and monitoring of these patients. So, our data require to be confirmed by further studies with a larger number of patients in a multi-center study. And thank you for your attention.